નમસ્કાર, હું આદર્શ છું. વેલકમ ટુ આદર્શ એજ્યુકેશન સોસાયટી. સ્ટેન્ડર્ડ નાઇન્થ, સબ્જેક્ટ સાયન્સ, ચેપ્ટર 5, the fundamental unit of life. See, dear students, what is fundamental? The fundamental unit of life means we are saying cell. Cell is the fundamental and essential part of the life. See, just we will go through the contents. So, what is there in these lessons? So, introductions of the cell, discovery of the cell, difference between unicellular and multicellular organism, difference between prokaryotic and eukaryotic organism, structure of the cell membrane, nucleus, cytoplasm, cell organelles, and lastly, difference between plant and animal cell. So these are the things we are going to learn in this topic. Okay. Now, just a small introduction, we'll go through it. So you know that what is cell? Cell is the fundamental unit of life. Why fundamental unit of life? Because you know that, see, starting with the cell, the group of cells we are called as tissues, and group of tissues are called as organs, and group of organs called as organ systems, and group of organ systems then produces organism. So it means the starting from the cell it starts. See, beginning is cell. So that is why the fundamental, so cell is considered as the fundamental unit of life. Okay, students. Now we'll discuss about the how cell was discovered. Remember that Robert Hooke in the year 1665, he discovered cell. How? He took a thin slice of bottle cork. You know that bottle, that cork, that is made of wood. Substance taken from the bark of the tree. The tree chalu bolte hai na, tree chalu. He observed that is made of cork. He observed studied self-designed microscope. He observed in the self-designed microscope. Like primitive, like old microscope. He saw that empty spaces bounded by walls, giving appearance of honeycomb-like structures. What he found? Small honeycomb rooms, just like honeycomb structures. Then this whatever he found a small room, that is we are calling box or compartments. That we are calling box or compartments. Okay, cell from the Latin word it is now known as cellula. It's a small room. And later, Hooke summarized, one more scientist, Hooke is summarized his, he also, later, Robert Hooke could not prove the, whatever the cell, but later Hooke summarized his all aspects in a book. He also could not do many things, but he has stored in his book Micrographia. The name of the book is Micrographia. Then in the later Robert Brown, he studied the cell in the 1833. That small dot appears in the cell. So that is called nucleus. And he found the nucleus in the orchid, which are decorative plants of the plant root. Then later studied Sheldon and Scavan. in 1813, all the living organisms are made up of cells. So they told all the living organisms are made up of cells. And further, Virtue in 1885 modified the definition of the cells. The cells are giving birth. So one cell is there, it becomes two cells, two cells is there, it becomes four cells, means giving birth. From the pre-existing cell, from the old one, or from the initial one. That means cell arises from the existing cells. So these are all short introduction about the cell. Now we will go through the detail about the line of the textbook. While examining a thin slice of cork, thin pieces of cork, Robert Hooke saw that the cork resembled the structure of the honeycomb structures of many little compartments, so many small rooms. And here given cork is a substance which comes from the bark of the tree. Tree bark, stem of the bark, chalu you are saying, that is comes from made from the tree, cork. That is a cap of the bottle. This was in the year 1665. So remember students, whenever many times you are studying but you don't know or not remembering the topic, what it tells. So sometimes asking who discovered the cell, then our answer should be in the year 1665, and who discovered? Robert Hooke. Made this chance observation through self-designed microscope. Old microscope. Okay, it is a self-designed microscope. Then Robert Hooke called these boxes cells. He called these boxes like small compartments. He called this cell. So cell in the Latin word, it means a little room. You know that in the honeycomb, small chambers or small structures, what you are saying, that is like a small room. This may seems to be very small and insignificant incident. Not given any importance, but it is very important in the history of the science. But those time, what he told Robert Hooke, not taken any so important, not given any importance, but it is very important in the history of the science. And this was the very first time that someone had observed that living things appear to consist of a separate units. Then some other one observed these living things appear separate units that we are calling the cell. The use of the word cell to describe these units is used till this day in biology, what you are calling about the given importance later about the cells. Now, what are living organisms made of? What are living organisms are made of? Okay, let us discuss about this. Clear? So let us take a small piece from onion bulb. You have to take the onion. Bulb means you have to take out the layer, outside skin, with the help of the forceps. Forceps is a thing which is used for, what you are saying? Forceps is used for holding the objects, at home, or this all we are using. We can feel off the skin, called epidermis. Epidermis means outer layer of the living beings, outer layer of the cell. From the concave side, concave and convex, the inner layer, concave side of the onion. This layer can be put immediately in a watch glass containing water. So last time in the video I have shown the watch glass also, okay, like a small vessel. This will prevent film from the getting folded. So that's why the means that film, thin slice, will not get folded or not get dry. So that is why you have to keep in the watch glass and you have to put in the water. Let us take glass slide. Then prepare a glass, in a small glass slide. Put a drop of water on it and transfer a small piece of the peel from the watch glass to the side. So from the watch glass you have to take and you have to keep on the glass slide. Make sure that the peel is perfectly flat on the slide. So it should be correctly placed on the slide. Then you have to use for this, a thin camel hair paintbrush might be necessary to help transfer. So here given for transferring for takeout, use that which one? Small camel hair paintbrush. Now we put a drop of iodine solution. On that you have to put iodine solution. Why? Because it gives color, so you can study the cell properly in the microscope. On this piece followed by the cover slip, and you have to put the cover slip also. Take care, avoid air bubbles. So that is why you are placing the cover slips, and avoid air bubbles should not be entered. So that we can find the structures easily. While putting the cover slip with the help of the mounting needle, mounting it like placing one above the other, like mounting needle. Ask your teacher for the help. We have prepared a temporary mount of onion peel. We can observe the slide under the low power followed by the high powers of compound microscope. So you have to see the compound microscope. Okay. See the structure here. So this is a eyepiece. This one is body tube, and this one is this one for adjustment, and this one is fine adjustment, and this one is objective lens what we are keeping, and this one is the stage, and this one is the shivel, and this one is mirror. This is called base, and this one is calling condenser, and here clip will be there for fixing and slide. So these are the what you are saying arrangement of the microscope. Now what you have to do? What you have to observe? What do we observe as we look through the lens? So what you will see? Then can we draw the structure? What we are seeing? Then you have to take what you are taking notebook page, and you have to draw that. See through the microscope and observation sheet, you have to draw it. Does it look like? You have to see that. We can try preparing temporary mounts of the onion swiss of different sizes. What do you observe? With similar structures or different structures. Now, what are these structures? So, this much your preparations will be there. These structures look similar to each other. See, what is the structure you are observing in these structures? We do these structures, and these structures we can preparing temporary mounts of the peels of onion, different sizes. So, you have to take a different size of the onions. The big one and smaller, what do you observe? Asking, what do you observe? Do you similar structures or different structures? If you take big and small size, whether they feel similar structures, so this you have to study. Then what are these structures? These structures look similar to each other. Whether you take the big onion, whether you take a small onion, but all the structure seems to be same. Together they form a big structure like an onion bulb. We find from this activity that onion bulbs of different sizes have similar structures visible under the microscope. The cells of the onion field will all look at the same, regardless of the size of the onion they came from. Whatever the size you take, but it looks similar shape. And these small structures that we see are the basic buildings. See what you are seeing? These are the basic buildings, units of the onion bulb. So these are basic, all the small structures are the basic units of how we are making, how we are constructing the wall. Small bricks are the basic units of the building. So these structures are called as cells. Not only onions, but all the organisms that we observe around are made up of cells. So all the organisms, all the living things are made up of cells. However, there are also single cells that live on their own. So they live on their own. Now we'll see the yellow colored box. Cells were first discovered by Robert Hooke in the 1665. He observed the cells in a cork slice with the help of the primitive, old microscope. Later, Leeuwenhoek in the 1674, with the help of the improved microscope, discovered the free living cells in the pond water for the first time. See here the question asking, who discovered the living cell? So then you have to say Leeuwenhoek. Robert Hooke discovered the cell, that is a dead cell. Okay, who discovered the cell? Then 1665, then Robert Hooke. But who discovered the living cells? So then you have to say Leeuwenhoek. In the year 1674, from the pond water, he observed some living cells. It was Robert Brown in the 1831, then later 1831, who discovered nucleus. The cell, he found in the center, nucleus, small dots. Later, Purkinje in the 1839 coined the term protoplasm. Surrounded the nucleus, found small liquids like structure, fluid substance of the cell. The cell theory that all the plants and animals are composed of the cells, and the cell is the basic unit of the life. So in this way, all the living organisms are, either it may be plants or animals, are made up of cells. Okay, this is the basic unit of the life. And later, it was presented by the two biologists, Sheldon in 1838 and Scaven in 1839. They told all the living bodies, either it may be plants or animals, body is made up of living cells. Then the cell theory was further extended by Virchow in 1855 by suggesting that all cells arise from the free existing cells. See, from the one cell making two cells, from the two cells going for four cells. So like this, it will divide, giving birth from the free existing, from the previous cell. So means from starting with the cell. With the discovery of the electron microscope in 1940. So it was possible to observe and understand the complex structure of the cell and its various. So due to the electron microscope, we can observe now even the complex structure also. Okay students, thank you. Thank you for joining us.